periphery of the cells, indicative of poor turnover and therefore low motility. While if you get rid of P63, there's this punctuated pattern of staining on the leading edge of the cell, indicative of more rapid turnover and therefore enhanced motility. Next, we wanted to ensure that it is delta NP63 alpha and not another isoform that is responsible for mediating these effects in MCF10A cells. So what I did this time is that MCF10A cells are human cells. So I knocked out all P63 isoforms, and then I reintroduced specific mouse isoforms that were MIC type, so it's a little bigger than this one. And the mouse and the human proteins are about 98 to 99.5% homologous. However, the mRNA is different enough that the short hairpin does not target the mouse messenger RNA. And then I took these cells and I subjected them to invasion assays. And I found that only the delta NP63 alpha, but not the delta NP63 gamma or the TA isoforms, could bring invasion back down after being elicited by knocking down P63 down to basal levels, demonstrating that in this system of MCF10A cells, delta NP63 alpha is the major isoform P63 that inhibits cell invasion. So now we were really happy, right? We had shown that reduced endogenous delta NP63 alpha downregulates MKP3, upregulates ERK signaling, and increases cell migration and invasion. So we wanted to know, well, is this increase in ERK signaling important for mediating these effects, right? And in order to tackle this question, first I did the easiest thing, and I simply inhibited ERK phosphorylation with a pharmacological inhibitor U0126, which is a MET inhibitor. And then I subjected the cells to cell invasion assays. And I found that in the presence of this U0126 compound, cell invasion after being elicited to increase by P63 knockdown was brought all the way back down to basal levels, showing that P63 ablation-induced cell invasion requires ERK signaling, right? We wanted to look deeper into this, right? We wanted to assess the role of ERK1 and ERK2 separately. So I decided to knock down either ERK1 or ERK2 or both, ERK1, ERK2 or both, in a background of P63 ablation, and then subject all these conditions to cell invasion assays. And I found, surprisingly initially, that when you get rid of ERK1, there's basically no change in cell invasion, right? ERK1 is not part of this. But when you get rid of ERK2, or both ERK1 and ERK2, cell invasion goes all the way back down to basal levels, showing that ERK2, but not ERK1, is absolutely required for mediating the effects of P63 reduction on cell invasion. We found similar results in terms of focal adhesion turnover. Control cells either with ERK expression or without ERK expression exhibited these large focal adhesion complexes around the periphery, just like I showed you before. When you get rid of P63, find a punctuated pattern of staining in the leading edge of the cells, and that is not affected when you get rid of ERK1. But if you get rid of ERK2 or both, then the staining pattern goes back into forming large complexes around the periphery, just as we expected from before. And then finally, we wanted to assess the direct contribution of MKP3 to mediating these effects. So this time, I overexpressed MKP3, I added exogenous MKP3 into a background of P63 ablation. And then I subjected these cells to cell migration and ablation and invasion assays. And I found that in both migration and invasion, adding MKP3 back in significantly or fully reverts P63 ablation-induced cell migration or invasion back to basal levels in invasion and significantly lower for cell migration, showing that MKP3 is at least partially required for these effects on cell migration and required for these effects on invasion. So just to recapitulate where we are now, we have found that delta MP63 alpha can upregulate MKP3 and exert negative regulation on ERK2, thereby inhibiting cell migration and cell invasion. But still, we wanted to know whether P63 is a metastasis inhibitor, right? And this is all great, but it's all in vitro, right? So we wanted to know, does reduced delta MP63 alpha affect cancer metastasis in vivo? So in order to answer this question, we used a well-established model of lung metastasis in which you get tumorigenic cells, inject them into the lateral tail vein of the mouse, of a nude mouse. Then the cells travel in circulation and get stuck in the lung capillaries because they're too big. So normally the cells would die, but if the cells are metastatic, they can infiltrate into the lungs and then grow as metastatic nodules, which then you can visualize after dissection. Right? There is one caveat, however, to this method, and that is that these cells need to be tumorigenic, and there are no breast cancer cell lines that are tumorigenic and that express P63, right? Arguably, because if they did express P63, then they wouldn't be tumorigenic, right? It's just my opinion. But so then, what we decided to do with the help of Michael Sherman and especially Meng Lo in Michael Sherman's lab, we transformed MCF10A cells, which do express P63, with an oncogene. So we transform them with HER2 mutant, which is denominated NUT, and we assessed neoplastic transformation by loss of contact inhibition and the formation of this foci when grown in monolayer. We found that NUT expression markedly increased phosphorylated ERK levels, as would be expected since it's an EGF receptor. But when we get rid of P63, we still observe the results as before, that MKP3 is lower, phosphorylated ERK levels are higher compared to cells that do express P63. And when we test this for in vitro invasion, we find that ablation of P63, even in the presence of such a potent oncogene, increases cell invasion. So we were happy, and I went ahead and I finally injected the mice with these cells, waited 45 days, and then euthanized the mice and dissected their lungs. I found that in the majority of the mice that were injected with cells with ablated P63 expression, you could see this very noticeable metastatic nodules on the surface of the lungs, in contrast to this healthy-looking lungs from the majority of the control mice. When we quantitated this, I found that only 1 out of 6 mice that were injected with control cells developed just 1 metastatic nodule on the surface of these lungs, while 3 out of 5 mice that were injected with P63 ablated cells developed 3, 4, and 5 metastatic nodules on the surface of the lungs. We wanted to confirm this, so we did an histological analysis which very clearly demonstrates cancer cell infiltration into the lungs when you compare them to the normal architecture of the lungs from the control mice, thus strongly demonstrating that P63 ablation decreases metastatic frequency in vivo. So that leads us to our working model in which delta MP63 alpha upregulates MKP3 to exert negative regulation ERK2, therefore inhibiting cell migration and invasion. But upon cancer progression, P63 levels decrease, causing a decrease in MKP3 expression, leading to an increase in ERK2 activity, which drives focal adhesion turnover, matrix metalloprotease 1 and 9 expression, increased cell migration, increased cell invasion, which then ultimately lead to metastasis. So with that, I just want to acknowledge some people